Welcome to The Deep Dive. We're the show that, well, aims to cut through all the noise and really deliver those key insights, those moments that genuinely help you feel well-informed. Today, we're plunging into a world that I think for most of us feels a bit mysterious, maybe even a little scary. The viral world. We tend to think of viruses as these invisible enemies, right? The things behind pandemics or just a common cold. But what if that common view is just, scratching the surface? What if it's way more complex? Honestly, pretty surprising. Like, what exactly is a virus? Is it actually alive, or is it something else, something kind of in between? And could it be that these tiny things aren't just about destruction? Could they also be hidden architects of life, shaping us, even tools we can use?
That's a perfect way to frame it. We're definitely going to challenge some of those common ideas today. Our goal here is to really unpack the true nature of viruses. We'll look at their origins, which are, frankly, mysterious, and really push the boundaries of biology. We'll cover their incredible diversity, how they're classified, the clever ways they spread, and maybe most surprising for many, we'll dive into their beneficial roles in nature that are critical for ecosystems, even for our own bodies. And then, we'll totally shift gears and look at how we're using them now, groundbreaking stuff in medicine, in pest control. It's not just about disease, it's about seeing this whole microscopic realm in a totally new light. We've dug through a pretty comprehensive stack of sources to pull out the really important stuff, giving you a shortcut to getting informed on this because it is often misunderstood.
Okay, fantastic. Let's start right at the basics then. When we say virus, what are we actually talking about? Because it's definitely not a cell, right? You don't picture them with little organs inside like a bacterium or one of our cells. They're different.
That's the absolute key difference right from the start. Fundamentally, a virus is just a small piece of genetic information. It could be RNA or it could be DNA. And it's housed inside this protective protein shell. We call it a capsid. And you're spot on. They aren't made of cells. That means they lack all the internal machinery. The ribosomes, mitochondria, all the energy-making stuff that cells have to copy themselves. Instead, viruses are basically just biological blueprints, very efficient instructions. Once they get inside a host cell, they hijack that cell's equipment, its machinery, and force it to make more copies of the virus. Think of it like a minimalist instruction set meeting an existing factory rather than being the factory itself.
So just the instructions and the protective shell, like a tiny self-assembling package. Is there anything else to their structure, though? Anything that helps them out?
Yeah, that's a great question because sometimes, yes, there is more. Beyond that basic capsid, which when you combine it with the genes inside, we call the nuclear capsid, some viruses have an extra outer layer. It's a lipid bilayer membrane, basically fat molecules, usually called an envelope. And what's really clever about this envelope is the virus doesn't build it itself. It actually steals it. It acquires this membrane by budding through one of the host cell's own membranes on its way out. Could be the main cell membrane, the nuclear membrane, maybe even the Golgi apparatus inside the cell. By wrapping itself in a piece of the host's own stuff, it achieves this kind of biological camouflage. It helps the virus sort of cloak itself, evade immediate detection by the host immune system. It looks like self, at least for a bit.
Whoa, that cloaking is incredibly smart, using a piece of the host against itself, almost like a tiny Trojan horse. So given all this cleverness, how small are we talking? When you say tiny, it's still hard to picture the actual scale.
Oh, they are incredibly small, almost unimaginably so. Typically, viruses range from about 20 nanometers up to maybe 400 nanometers.
Yeah.
To put that in perspective, the smallest ones are like 2,000 times smaller than a grain of sand. And compared to the cells in your body, they're roughly 100 to maybe 1,000 times smaller. And even within the virus world, there's a big size difference. Like the measles virus is about 5 times larger than the Zika virus.
Yeah.
So it's a whole spectrum, even down at that microscopic level. And these protective capsids, they also have distinct shapes, distinct architectures. We call these symmetries.
Symmetries. That sounds very precise, very geometric. What are the main types? What do they look like? Does it matter?
Absolutely. One common type is helical symmetry. This gives you kind of rod-shaped virus. Picture like a spiral staircase or a spring. The genetic material winds up in a helix, and the protein subunits arrange themselves around it, making this long tube. The classic example is tobacco mosaic virus in plants. But in animals, you see it in familiar ones like influenza, Ebola, rabies. And interestingly, all the helical animal viruses we know are enveloped. They have that lipid cloak we talked about, whereas most helical plant viruses are naked. They don't have the envelope. Maybe different evolutionary needs there.
So the plant ones skip the cloak. What's the other main shape then?
The other really common one is icosahedral symmetry. This makes the virus look more sort of cubic or spherical. An icosahedron is a geometric shape. It's got 20 identical triangular faces, 12 corners. It's incredibly efficient. Think of it like a tiny self-assembling geodesic dome or a fortress built from these protein blocks to protect the genes inside. It's a really stable structure, maximum internal space for the genome, using the minimum number of protein parts. It's evolutionary genius, really. Lots of common human viruses look like this adenoviruses causing colds, poliovirus, herpes, simplex virus. They all use this super efficient, sturdy design. And the way these proteins are arranged is key for how the virus gets into cells.
That structural precision is fascinating. But okay, here's where it gets really interesting for me, maybe a bit philosophical. If they can't reproduce on their own, they don't have cells, they don't make their own energy, how do scientists even decide if something like that is alive? They seem to sit right on the edge, in this weird biological gray area.
It absolutely feels like a gray area. And yeah, this is a huge, long-running debate in biology. There's no simple answer. Scientists are still grappling with it. Are viruses living things? If you look at the standard checklist we use for life, viruses kind of mess it all up. They meet some criteria, but totally fail others.
Okay. Lay it out for us. Where do they fit? And where do they fall short on that traditional life checklist?
Yeah. Right. Let's break it down. First big one, having cells. All things we generally agree are alive are made of cells. Viruses, nope. As we said, no cells. Just genes and a protein code. So that's a big check in the not alive column.
Okay. No cells. What about reproducing?
Reproduction. Living things reproduce, pass on genes. Viruses do replicate. They do pass on their genetic info. Sounds alive, right? But they don't have the tools they need to reproduce on their own. They have to invade other cells and force them to make the copies. So yes, replication, but not independently. They're obligate intracellular parasites, dependent.
Dependent replication. Okay. What about energy, metabolism?
Yeah, making and using energy or metabolism. Living things take in nutrients, make energy. Viruses can't make energy. They're just inert, inactive until they get inside a host cell. Once inside, sure, they use a ton of energy for replication, but it's all stolen from the host cell. They're energy parasites, too.
No energy production on their own. Got it. And the last one, responding, adapting.
Right, responding and adapting to their environment. Living things interact, maintain balance, evolve. Viruses do interact with host cells, and they absolutely evolve, sometimes incredibly fast. They adapt through mutation, natural selection. But the sources note their response isn't really active like in other organisms. It's more of a passive chemical interaction until they bump into the right host cell. So they adapt over generations, but maybe don't respond moment to moment in the same way a bacterium might.
So not cellular, can't make energy, need a host to copy themselves. Sounds pretty non-living. But they replicate, they evolve, they have a massive impact on life. Sounds pretty alive. It really is a riddle. It pushes how we even define life.
It really is. It forces us to question that definition. Mm-hmm. Historically, you had ideas like Pasteur's life comes from life, omni vivo ex vivo, and concepts like vitalism that drew a sharp line between living and non-living. Viruses just blow that simple binary distinction apart. They blur the lines completely. What's fascinating, though, is even if it's debatable whether they fit our definition of alive, they are undeniably, and probably have always been, a huge part of life, part of the construction of life. They replicate, they evolve key features. Maybe for viruses, the question isn't so much what is life, but maybe how is life? How does it manifest in these different interconnected ways? They're like a different strategy for existence, completely tangled up with cellular life, but operating by different rules.
That's a really profound way to think about it. Not just alive or not, but part of a spectrum of how existence works. So if they're in this weird state and maybe super ancient, where did they even come from? Were they always here? Maybe from before cells? Or did they, come from somewhere else?
That origin question is one of the biggest mysteries. And when we say somewhere else, we're not talking UFOs. We mean potentially from a time on Earth before or alongside the very first cells. One major idea is the viruses without cells hypothesis, sometimes called virus first. This suggests viruses came about really early in the development of life, maybe even before the first proper cells. They might be leftovers from a primordial RNA world, that hypothetical time when RNA did everything, acting as both genes and enzymes, before DNA took over. And the evidence for this? Our sources point to the sheer profound diversity of genomic strategies revealed by the Baltimore classification, which we'll get into more detail on later. But basically, viruses use at least 7 fundamentally different ways to manage their genes, including using RNA genomes and that heretical process of reverse transcription making DNA from RNA, which goes against the usual flow.
So the fact that they operate so differently from our cells or bacteria, genetically speaking, suggests they're incredibly ancient, maybe from a time when life's rules were different.
Exactly. The argument is, if viruses had just escaped from modern cells later on, You'd expect their replication strategies to look a lot more like their hosts, following that standard DNA to RNA to protein path. The fact that they're so radically different suggests they are not mere offshoots of modern life, but represent a fundamentally different and far more ancient lineage. It's like finding an incredibly old, unique dialect that must have split off ages ago, pointing to some forgotten root language. They might be direct descendants of Earth's earliest self-copying molecules.
Wow, okay. Viruses as primordial life forms. Mind-bending. What's the other main theory? The escaped genes idea.
Right, the alternative is the viruses from cells, escaped genes hypothesis, also called the escape or maybe reduction hypothesis. This suggests viruses evolved from bits of genetic material genes that escaped from cellular organisms at different points in early evolution. These escaped bits, maybe DNA or RNA, then somehow gained the ability to replicate themselves semi-autonomously, selfishly, and infect other cells. So instead of being first, they're seen as later offshoots of cellular life that simplified, shedding everything they didn't need to become efficient parasites. They reduced themselves.
Okay, so one theory says they came first, the other says they broke off later. Is there any kind of middle ground, something that combines the...
Yes, there is. A more recent view gaining traction is the chimeric scenario. This tries to blend parts of both. It suggests maybe viruses got their core replication machinery from different types of primordial genetic elements, echoing that ancient RNA world idea. But then they obtained the structural proteins required for capsid formation from their cellular hosts, bar them, essentially. So it paints a picture of viruses as evolutionary patchworks, mosaics, grabbing bits and pieces from different sources at different times, ancient replicators and evolving cells.
A mix and match evolution. That makes intuitive sense. Is there anything structural or functional that links all viruses, suggesting some kind of deep shared ancestry regardless of which origin story is right?
There is actually a crucial unifying theme. Our sources highlight that the core replicative enzymes, the machinery viruses use to copy their genes across all the major viral groups, these enzymes are built on a remarkably conserved structure. It's called the core RNA recognition motif, RRM domain. This RRM domain is super common across all life. It's fundamental for handling genetic material. The fact that it's so central in viruses suggests that maybe all these different modes of replication emerged within the primordial pool of genetic elements, replicators, before cells even fully separated out. It hints that the basic tricks for copying genes might have originated in a very early shared stage of life's evolution, and viruses could be direct descendants or very close relatives of those first self-copying entities.
So these viral replicators might have been evolving even before the first cells we'd recognize, sharing a really ancient root with cellular life. That's foundational. And what about their history since then? Have they been shaping life all along?
Oh, absolutely. The constant back and forth, the evolutionary arms race between viruses and hosts suggests a really dynamic history. The sources mention that mass extinction of resident viruses might have happened historically on a huge scale, just like we see species go extinct. Dominant viral groups likely rose and fell as hosts changed or new viruses came along. It implies this deep coevolutionary dance has been going on for billions of years, shaping all life. Viruses aren't just passengers, they're active players, maybe even driving major evolutionary shifts.
Okay, that's a lot to take in. Tiny entities, maybe not alive, possibly older than cells, huge diversity. So given all that complexity, how on earth do scientists even start to organize them? What does the family tree of a virus look like? Do they even fit on a tree?
It's a massive challenge, precisely because of that diversity and their unique nature. The main group doing the formal organizing is the International Committee on Taxonomy of Viruses, the ICTV. They've been around since 1966. They're the sole authority for naming and classifying viruses. And the key thing is, because viruses aren't considered alive in the same way cells are, they get their own separate classification system. It doesn't connect directly into the standard tree of life for bacteria, plants, animals.
So it's not like kingdom, phylum, class, order, family, genus, species, or is it similar?
It uses similar ranks, but it's a bit streamlined, though expanding. Traditionally, it focused mainly on order, family, genus, and species. More recently, the ICTV has added higher ranks like realm, kingdom, phylum to handle the sheer scale of viral diversity they're discovering. And just like regular biology, the names are in italics. Taxa above species have specific endings, like families ending in viridae, orders in viral lines. Species names are binomial, like genus plus a species epithet, similar to Homo sapiens.
Can you give us an example of how this taxonomy works in practice? Maybe how it changes as we learn more.
Sure. A really good example is the rubevirus genus, which includes the rubella virus cause of German measles. For decades, since '75, rubevirus was parked in the Togaviridae family. But as we got better at sequencing genomes, scientists realized rubiviruses were actually quite different from other togaviruses, like alphaviruses. Differences in how they spread, their variant structure, their gene sequences. So based on all this new data, in 2019, the ICTV moved rubivirus into a whole new family, Matonaviridae. It shows taxonomy isn't set in stone. It evolves as our knowledge grows, leading to more accurate groupings.
Okay, so that's the formal hierarchical system from the ICTV. You mentioned another system earlier, the Baltimore classification. How does that fit in? Is it competing or complementary?
It's definitely complementary, not competing. While the ICTV focuses on the evolutionary hierarchy, the Baltimore classification system, from Nobel laureate David Baltimore, is more of a functional classification. It brilliantly organizes viruses based on something fundamental: how viruses produce messenger RNA, mRNA, and replicate their genomes. This tells us a huge amount about their basic biology, their internal operating system, how they actually work inside a host cell. It groups all viruses into 7 major classes, each with a distinct strategy for managing its genetic information and making the mRNA needed for viral proteins.
7 classes. Okay. Can you quickly run through them? What makes each strategy different?
Sure, the 7 classes cover all the known viral strategies. Class 1. These are dsDNA viruses, double-stranded DNA. Their genome is DNA, just like ours, and it gets transcribed into mRNA. Herpes viruses, pox viruses are examples. Class 2, ssDNA viruses, single-stranded DNA. Their single DNA strand has to be converted to double-stranded first, then transcribed. Think parvoviruses. Class 3, dsRNA viruses, double-stranded RNA. Having dsRNA is unusual. They use their own enzyme to make mRNA directly from this dsRNA template. Rotaviruses fit here. Class 4, positive sense ssRNA viruses plus ssRNA. Their RNA genome is ready to go. The host cell's ribosomes can read it directly like mRNA to make proteins immediately. Picornaviruses like colds, polio, flaviviruses, Zika, dengue, togaviruses. Class V, negative sense ssRNA, they're RSC in a sense. It can't be read directly. It must first be transcribed into a positive sense mRNA copied by a viral enzyme. Influenza viruses, orthomyxoviridae, measles, mumps, paramyxoviridae, rabies, rhabdoviridae are big ones here. Class 6 RNA viruses that reverse transcribe. These are the famous retroviruses. They have a plus ssRNA genome, but they carry an enzyme, reverse transcriptase, to make a DNA copy of their RNA. This DNA copy then gets inserted into the host's own DNA. HIV is the classic example, class 7. DNA viruses that reverse transcribe. These guys have a DNA genome, but they still use reverse transcriptase at a certain stage in their replication cycle. Hepatitis B virus is the key example here.
Wow, that is an incredible range of genetic tricks for such tiny things. It really hammers home that radical diversity you mentioned about their origins. Do these different strategies line up with the kinds of hosts they infect? Like, do bacteria mostly get DNA viruses and animals RNA viruses?
They absolutely do, and it reveals fascinating evolutionary patterns. When you look at the host range distribution across these classes, you see clear trends. There's a real dominance of dsDNA viruses in bacteria and archaea, the simpler prokaryotic cells. This strongly contrasts the dominance of viruses with RNA genomes in eukarya, that's us, plants, fungi, protists, the more complex cells, and things like RNA and dsRNA viruses being nearly confined to eukaryotes. It suggests different strategies might be better suited for different types of cellular environments, maybe reflecting different host defenses or cell structures they evolved alongside.
What's fascinating here is this Baltimore system, it's like understanding their core operating system, isn't it? Not just what they are, but how they fundamentally work. It gives us clues about their history, evolution, and how they mess with different kinds of life. Okay, so we've got a handle on what viruses are, their weird alive or not status, their ancient roots, and how we classify their amazing diversity. But let's bring it back to the practical side. How do these tiny, complex things actually get from one host to another? How do they spread? What are the main ways?
Right, that's the crucial link to disease and public health. Viruses can pass from person to person in various ways, leading to everything from the mild common cold, rhinoviruses, adenoviruses, to really life-threatening diseases like Ebola. And the way they transmit isn't accidental. It's key to their survival and evolution.
So what are some of the main routes we encounter? Like, how does a cold virus actually spread?
One of the most common is simple direct or indirect contact. Think about those cold viruses, rhinoviruses and adenoviruses. They have tough protein coats. And interestingly, they're not necessarily deactivated by soap like some bacteria are. But soap is still super effective because it physically lifts them off surfaces, including our skin, dislodging them. so that they can be washed down the sink. It's
Mm-hmm.
physical removal. That's why our sources emphasize washing your hands with soap and water is better than using a hand sanitizer in many cases. Sanitizers kill some things, but they do not have the same effect of removing the viruses from our skin like good old soap and water washing does.
So it's not about killing the virus with soap. It's literally washing it off. That's a really important distinction people might miss. Are there more sort of stealthy or efficient ways they spread within an organism once they're in?
Absolutely. A highly efficient method is cell-to-cell transmission. This can happen through structures sometimes called infection synapses. Think of them as direct, intimate connections between adjacent cells. The virus passes directly from one cell to the next without ever having to go out into the surrounding fluid. This method is estimated to be way more efficient, maybe 100-fold more efficient than the transmission of viral particles released into the extracellular environment. It's like passing a note directly instead of shouting across the room. It allows rapid local spread and can help the virus evade immune cells patrolling outside the cells. HIV uses this effectively.
Wow, a direct connection, bypassing the defenses. Very clever. What about involving other creatures, like insects carrying diseases?
Exactly. That's arthropod-borne transmission, and it's huge for global health. The fact that infectious cells can be disseminated by insects provides a model for an early origin of arthropod-borne viruses. This is how diseases like Zika, dengue, West Nile virus are spread. An insect vector, like a mosquito or tick, picks up the virus from one host and injects it into another when it bites. The virus often has to replicate in both the insect and the mammal or bird host to keep the cycle going. It's a complex lifestyle.
So these different ways of spreading contacts, cell-to-cell insects, they aren't just random, are they? Do they shape how the virus evolves? Like, does a virus that spreads easily become less deadly?
They are absolutely fundamental drivers of evolution. The pathways of viral transmission are intricately linked to their evolutionary success. And often this favors strategies that moderate virulence to maximize spread. Think about it. If a virus kills its host really quickly, the host might not get out much, might not encounter many new potential hosts. That limits the virus's spread. A virus that causes milder symptoms, allowing the host to stay mobile and interact with others for longer, might actually spread much more effectively. So there's this evolutionary trade-off. The most successful viruses often strike a balance effective enough to replicate, but not so deadly they burn out their transmission opportunities too fast. And this really brings us to a key point. Understanding these transmission mechanisms is vital, not just for stopping diseases, but for appreciating this constant evolutionary dance between virus and host. It shows how viral success is often about persistence and spread, sometimes achieved through surprisingly subtle means, leading to long-term coexistence, not just destruction.
Okay, let's shift gears then. Because when we think virus, the word dangerous just pops right into mind, doesn't it? Outbreaks, illness. But you're saying there's a completely different side to this, that some viruses might actually be good or helpful. That feels totally counterintuitive.
It absolutely does challenge the stereotype, but yes, it's a major shift in thinking. Viruses are being redefined as more than just pathogens. Increasingly, we're seeing them as critical symbiotic partners in the health of their hosts. This isn't just a niche idea. It applies across the whole tree of life. They can be silent collaborators.
Symbiotic partners, how does that work? What does that mean beyond just causing disease?
Well, symbiosis just means organisms living together closely. It can be mutualism, good for both, commensalism, good for one, neutral for the other, or parasitism, good for one, bad for the other. And our sources note that symbiotic relationships can lead to symbiogenesis. That's the amazing idea that two separate entities can fuse over time to create something new, a new species or biological system. And the evidence, the extent of virus-like sequences in the genomes of just about everything. It's proof of viral symbiogenesis that has shaped modern genomes. Viruses haven't just infected us, they've literally become part of our genetic code, sometimes adding new functions over evolutionary time. They're woven into the fabric.
Whoa, okay. They've helped build us genetically. That's massive. Can you give some specific examples of these beneficial relationships, not just in humans, but across nature?
Definitely. Let's start small, with viruses of microbes. Killer viruses of yeasts and bacteria, for instance, can actually help their hosts. They might produce toxins that kill off competing yeast or bacteria strains, allowing their host to take over a new territory. The host carrying the virus gets immunity to its effects, while its competitors get wiped out. It's like microbial warfare where the virus is the weapon.
Okay, helping their host compete. What about bacteriophages, the ones that infect bacteria? You mentioned they're dominant in our gut.
Right, phages. They're incredibly diverse and can do amazing things for their bacterial hosts. They encode essential functions for bacteria. For example, Some phages carry the genes for toxins that make an otherwise harmless bacterium pathogenic to us or other animals. The toxin allows the bacterium to invade its macrohost. They're also key players in horizontal gene transfer, moving genes between bacteria, maybe genes for antibiotic resistance or other useful traits. And some phages even help bacteria form biofilms, those resilient slimy communities that are hard to treat. So phages aren't just predators, they can be crucial allies for bacteria, shaping entire microbial communities.
Okay, so they can help bacteria become more dangerous or harder to kill, which, from our perspective, maybe isn't good. What about viruses helping other microbes that aren't bacteria?
That's a fair point. Beneficial is definitely relative to the host. But yes, viruses infecting other eukaryotic microbes, fungi, protists can also have positive effects on the growth, fecundity, or persistence of their hosts. Like a virus infecting a fungus that lives inside a plant might help the fungus, and indirectly the plant, tolerate heat stress or resist other diseases. But maybe the most stunning examples come from the giant viruses. These things are huge for viruses, almost bacterial-sized with massive genomes. They really blur the lines. They have hundreds, even thousands of genes, including ones for things like metabolism or making proteins, functions normal viruses totally outsource. Giant viruses. Blurring the lines, is there a specific example of one doing something really unexpected, something truly mutualistic? There's an amazing one, a giant virus that infects a marine protist, a tiny choanoflagellate. This virus has a huge genome, nearly 1 million base pairs, and it includes genes for 3 rhodopsin-like proteins. Rhodopsins are light-sensitive, they can absorb light and pump protons to generate energy. It's like a form of light harvesting, photoheterotrophy. And here's the kicker. The virus also contains the entire metabolic pathway needed to synthesize the retinal pigments required for these rhodopsins to function.
Hold on. The virus has the genes to capture light energy and the genes to make the specific molecules needed for that process, and it gives this ability to its host. That's unbelievable integration.
Exactly. It suggests the virus isn't just a parasite, it's a metabolic partner. It might be giving its host the ability to supplement its energy intake using light, maybe helping it survive in nutrient-poor waters. It's like the relationship between corals and their photosynthetic algae, but with a virus providing the light harvesting machinery. It really shows viruses can be agents of biological innovation. So if you connect this to the bigger picture, it flips the script. Maybe symbiosis and mutualism have been the norm for viruses throughout evolution, and the killer personalities are just the ones we notice most. They might be ancient collaborators more often than destroyers.
That is incredible. So viruses aren't just outsiders attacking, they're inside us, shaping our biology, even giving superpowers to other organisms. How deep does this go in humans? Are they really part of our makeup?
Oh, incredibly deep. Right into our DNA. The prime example here is endogenous retroviruses, or ERVs. These are basically vestiges of the continual bombardment of their host genome by exogenous retroviruses during evolution. Think of them as fossil viruses. Ancient retroviruses infected our ancestors' sperm, or egg cells, inserted their DNA copy into the genome, and then got passed down through generations. They became inherited.
Genetic fossils of old infections. Wow. But are they just sitting there like historical markers, or do they actually do anything beneficial for us now?
That's the amazing part. They're not just inert fossils. Our human genomes have something like 400,000 ERV locations. Now, that's just a tiny fraction that survived natural selection and became fixed in the population. Most insertions were probably harmful or neutral and got lost, but some are incredibly ancient. Endogenous hepatitis B viruses, EHBVs, have been found in animal genomes over 207 million years old. Their sheer persistence suggests they must be doing something useful, otherwise evolution likely would have weeded them out.
Okay, ancient, widespread in our DNA. What are these beneficial jobs they're doing?
Their roles are surprisingly diverse, mostly centered around gene regulation. The key parts are the long terminal repeats, or LTRs, at the ends of the integrated viral DNA. These LTRs naturally contain promoters and enhancers transcription factor binding sites. These are the switches and dials that control when and where our own genes get turned on or off. And our cells have essentially exploited these built-in viral control elements for our own purposes, co-opted them.
So they act like dimmer switches or specific on buttons for our genes, maybe only in certain tissues. That's like nature's genetic engineering.
It really is. For instance, one specific HRVE LTR acts as a parotid-specific enhancer for the gene-making amylase, the enzyme that digests starch. That helps ensure it's made specifically in our salivary glands. Another one, an ERV9 LTR, promotes testis-specific expression of a gene called P63, crucial for development in males. LTRs also heavily influence placental expression of several human genes. The placenta is a really dynamic organ, and ERV-LTRs can act as alternative promoters, providing different start sites for genes, sometimes giving them new or altered tissue specificity. Occasionally, an ERV-LTR even becomes the primary or sole promoter for a gene, completely taking over its control. And they can also provide polyadenylation sites, crucial signals for finishing off an mRNA molecule correctly.
So they're not just flipping single switches. They might be involved in rewiring whole circuits, especially during development.
Exactly. They're implicated in whole regulatory networks. For example, certain mouse ERVs act as promoters for many genes, specifically in early embryos, suggesting they may participate in reprogramming of the embryonic genome, setting up the initial patterns of development. This extends even to fundamental cell identity. A specific type, HRVH, and its LTR7 promoter drive transcripts that are abundant in human embryonic stem cells. This suggests they play an important regulatory role in human stem cell identity and maintenance of pluripotency, that ability to become any cell type. Our stem cells might rely on these ancient viruses. And finally, ERVs can also make non-coding RNAs that regulate genes in other ways, adding yet another layer of control. What's truly fascinating is this pattern. New retroviral insertions are often bad news, pathogenic. But the ones that survive over evolutionary time, that avoid being harmful or getting lost, can become neutral or even actively beneficial. They get harnessed by the host, fundamentally shaping how our genes work and how our genomes evolve. It's a stunning example of turning an ancient invasion into a vital partnership.
That is genuinely profound. From invader to essential component of our own instruction manual, it completely changes how you think about viruses. Okay, so beyond these natural, evolved partnerships, can we actively use viruses now? For specific goals like controlling pests that damage crops or spread disease, it seems like the next logical step.
We absolutely can, and it's a really active area. One major field is biocontrol, using biopesticides. These are often seen as a greener alternative to synthetic chemicals. They tend to be cheap, environment-friendly, specific in their mode of action, sustainable, do not leave residues, and are not associated with the release of greenhouse gases in the same way chemical production might be.
Greener, more targeted pest control. What kinds of viruses are used, and how do they actually work?
We mainly use microbial pesticides, derived from microbes including viruses. A key group are the baculoviruses, Baculoviridae. These are great because they are pathogens that attack insects and other arthropods, but are generally harmless to plants, humans, and other animals. Very specific. They typically have double-stranded DNA genomes protected by a tough protein coat called a polyhedron. The insect has to eat the virus. Once ingested, the virus infects the insect's gut cells, replicates massively, and the infection is typically fatal. The insect often liquefies, releasing billions of new virus particles back onto the plants to infect more pests.
Gross, but effective. The insect eats the virus, becomes a virus factory, and then spreads it more. Are there commercial products using this?
Yes. For example, GP Check uses a baculovirus, the gypsy moth nucleopolyhedrosis virus, to control gypsy moths, which can devastate forests. The advantages are clear. Highly specific in action, usually not harming beneficial insects like bees or predators. They can be easily sourced or produced, and they're environmentally sustainable without residual effects. This is a big contrast to some synthetic pesticides that can build up in the food chain, biomagnification, or accumulate in animal tissues, causing long-term harm.
That sounds much better for the environment and health. Are there downsides? Why aren't they used for everything?
There are some practical limitations. They often have a short shelf life compared to chemicals and break down relatively quickly in sunlight or soil, less persistent. So you might need to apply them more often or time it carefully. And their specificity, while great for safety, can be a drawback if you have multiple different pests. You might need a different virus for each one rather than one broad-spectrum spray. It requires more precise knowledge of the pest.
Okay, makes sense. How does regulation work for this? The EPA in the U.S. must oversee using viruses as pesticides, right? Especially if we're talking genetically modified plants producing viral components.
Oh, yes. The EPA regulates this very carefully. They oversee biochemical pesticides, microbial pesticides like baculoviruses, and also that category you mentioned, plant-incorporated protectants, or PIPs. PIPs are defined as pesticidal substances that are produced in a living plant, along with the genetic material necessary to produce the substance. The classic examples are beet varieties of crops, corn, cotton, potato, engineered with a gene from the bacterium Bacillus thuringiensis to produce an insecticidal protein. These target specific pests that eat the plant.
So the plant makes its own insecticide. Ingenious. What kind of safety checks do these engineered plants go through?
Very rigorous checks. The EPA looks at potential mammalian toxicity, allergenicity, effects on non-target organisms like butterflies or beneficial bugs, and environmental fate, how long the substance lasts. A big concern is gene flow, the risk of the engineered gene escaping from the crop into wild relatives. For instance, while Brit corn and potatoes seem low risk for gene flow in the U.S., Brit cotton sales are restricted in places like Hawaii and Florida because wild cotton relatives exist there, and gene transfer is a possibility they want to avoid. It's about careful risk assessment based on the local ecology.
That makes sense, managing the risk of spread. What about the insects themselves? Do they become resistant to these Brit plants, just like they do to chemical sprays?
Absolutely. Insect resistance is a major challenge. That's why insect resistance management, IRM programs, are mandatory for beet crops. A key strategy is the high-dose refuge approach. The plants produce a very high dose of the beet toxin, much more than needed to kill susceptible insects. Then farmers are required to plant structured refuges nearby areas of the same crop without the beet gene. These refuges maintain a population of susceptible insects. If any rare resistant insects survive on the beet crop, they're likely to mate with the abundant susceptible insects from the refuge. This dilutes the resistance genes in the population, slowing down the evolution of widespread resistance. It's an ongoing management strategy.
A clever ecological strategy to slow down evolution. Is there a big, real-world example of using a virus for long-term pest control, showing this evolutionary back and forth?
The classic case study is Australia's rabbit biocontrol program. Rabbits were introduced in the 1800s and became an ecological nightmare. In 1950, they deliberately released the myxoma virus. It causes myxomatosis, which was initially incredibly lethal, killing maybe 99% of infected rabbits. Huge population crash. But over time, what happened was coevolution. The virus evolved to become less deadly, and the rabbits evolved to become more resistant.
Wait, the virus became less deadly. Why would it do that?
Because a virus that kills too quickly doesn't spread as well. If the rabbit dies in a day, it doesn't have much time to infect other rabbits. Virus strains that kept the rabbit alive longer, even if still sick, had more opportunity to transmit. So selection favored slightly less virulent strains. At the same time, rabbits with some natural resistance survived better and passed on those resistance genes. It's a perfect textbook example of the coevolution of virus virulence and host resistance, a biological arms race. Because rabbit numbers eventually recovered somewhat, Australia later introduced other viruses like strains of rabbit hemorrhagic disease virus, RHDV, in an ongoing effort. It's a dynamic, evolving control strategy.
So what does this all mean? We're essentially deploying nature's own highly specific weapons to manage ecosystems and food supplies, but it's a constant balancing act with evolution. We have to keep adapting our strategies as the pests and the viruses adapt too. That's fascinating for environmental control. But now the really mind-bending part. If viruses can control pests, can they actually heal us? Can we turn them into medicines? It sounds so paradoxical.
It does sound like a paradox, doesn't it? But the answer is a definite yes. It's one of the most exciting frontiers in medicine. Viruses are becoming indispensable medical delivery vehicles, often called viral vectors, especially in the revolutionary field of gene therapy.
Viral vectors, delivery vehicles. How do you take something known for causing disease and make it deliver a cure?
It's ingenious, really. Researchers basically gut the virus, taking out its harmful parts while keeping its amazing ability to get inside cells. They disarm the virus by removing some or all of its genome, the bits that cause disease. They might also tweak capsid proteins on the outside to make it less likely to trigger an immune reaction or to help it target specific cells, like liver cells or nerve cells. Then they fill the hollowed-out capsid with whatever they want to insert into cells. That payload could be a healthy copy of a defective gene for treating genetic disorders. It could be genome editing machinery like CRISPR to make precise DNA repairs. It could be a therapeutic drug, or even components for a vaccine. The virus becomes a highly efficient, naturally evolved nanoparticle delivery system.
Like microscopic delivery drones programmed for specific cells, carrying genetic cures. What kind of treatments are being developed or used with this?
The range is growing incredibly fast. For conditions needing permanent genome changes, like fixing the gene defect in sickle cell disease, modified lentiviruses and retroviruses are often used. They're good at integrating the therapeutic gene into the host cell's DNA for a long-lasting effect. Then there are CAR T-cell therapies for cancer, which are transforming treatment for some leukemias and lymphomas. Doctors take a patient's T-cells, use viral vectors to deliver a gene that helps the cells recognize and attack that cancer, grow millions of these engineered T-cells, and infuse them back into the patient. You're weaponizing their own immune system. Researchers are also constantly refining the vectors, making liver-sparing AAV capsids, AAVs are adeno-associated viruses, popular vectors, to avoid toxic buildup in the liver when high doses are needed. Safety and precision are huge focuses, and viral vectors are key for developing new vaccines against tough targets like Zika virus and malaria parasites. Even bacteriophages are being explored for vaccine delivery, like for COVID-19 in early studies.
That potential is just staggering. A true medical revolution. But it can't be easy. What are the big hurdles? The challenges?
Oh, definitely significant challenges remain. A big one is the immune system. Even if the virus is disarmed, the capsid proteins sometimes instigate an immune response, which can neutralize the vector or cause side effects. There's also limited cargo space. Viruses are tiny, you can only fit so much genetic material inside, and often low uptake means patients must receive massive doses to get enough vectors to the right cells, which increases risks and costs. And speaking of costs, they are astronomical, up to $2 million per treatment for some approved gene therapies. That raises huge questions about access and affordability. But beyond gene therapy, there's another really exciting use, oncolytic viruses, OVs, for treating cancer directly. These are viruses that naturally, or after being engineered, preferentially kill cancer cells and by helping the immune system do so. Their superpower is that they go after cancer cells while largely sparing healthy cells.
How do they know the difference? How do they target cancer cells but leave normal ones alone?
It often comes down to weaknesses in the cancer cells themselves. Many cancer cells have broken antiviral defense systems, like the interferon pathway we mentioned. So a virus like vesicular stomatitis virus, VSV, which healthy cells normally fight off easily using interferons, finds cancer cells are wide open targets. They become highly sensitive to infection with VSV. Once VSV gets in, it replicates like crazy, a rapid cytolytic replication cycle, killing the cancer cell and producing maybe a 1,000-fold amplification of virus within 24/8 to infect neighboring cancer cells.
So the virus turns the cancer cell into a factory for more cancer-killing viruses, causing it to burst. That's targeted destruction. Is this being used in patients? Are there approved ones?
Yes, there is one FDA-approved OV called TVEC, Talmogene Lahore-Paravec. It's a modified herpes virus used for advanced melanoma. It's injected directly into tumors. TVEC kills tumor cells, but maybe more importantly, it causes the dying cells to release signals that wake up the patient's immune system. It helps generate a systemic immune response against the cancer, potentially attacking tumors elsewhere in the body too. It acts like an in situ cancer vaccine.
Turning the tumor against itself immunologically. Are other viruses being tested like this?
Absolutely. Many different viruses are in clinical trials as oncolytics. Adenovirus, other herpes viruses, Maraba virus, interestingly an insect virus, measles virus, Newcastle disease virus, a bird virus, picornaviruses, Rio virus, vaccinia virus, the old smallpox vaccine virus. Each has unique properties making it suitable for different cancers or approaches.
And are scientists tinkering with these viruses to make them even better cancer fighters?
Constantly. There's directed evolution in the lab to breed better variants. There's transductional targeting, modifying the virus surface to stick specifically to cancer cells. There's non-transductional targeting, changing the virus's gene so it can only replicate inside cancer cells. And transcription targeting, putting crucial viral genes under the control of switches that are only on in cancer cells. And often, these OVs work best not alone, but in conjunction with conventional cancer therapies like chemo, radiation, or especially with immunotherapies like checkpoint inhibitors. They can work synergistically, hitting the cancer from multiple angles. Now let's switch to one last but hugely important area, phage therapy. Using bacteriophages, those viruses that eat bacteria, as an alternative or helper to antibiotics. Remember, phages are the natural predators of bacteria, and they're incredibly abundant, especially in our gut.
Right, the bacteria eaters. This sounds incredibly relevant now with antibiotic resistance being such a massive problem.
Exactly. As antibiotics fail more often, phages offer huge potential. Their big advantage is specificity. They typically inhibit specific species or strains of bacteria without causing any harmful effects on other strains. Unlike broad-spectrum antibiotics that nuke our good gut bacteria too, phages can be like sniper rifles, targeting just the bad guys. They're also often effective against bacterial biofilms, those slimy, antibiotic-resistant shields that cause so many chronic infections. Phages can sometimes chew through the biofilm slime.
Specificity and biofilm busting. That sounds ideal. Aren't there real-world successes, even in people?
Lots of promising examples. Phage P100 can clean listeria off foods. Phage therapy has reduced C. diphtheral colonization in biofilms. It helped tackle an E. coli outbreak in Germany. It works in animals too, protecting fish, mice from S. aureus, Vibrio, drug-resistant E. faecium. And yes, human trials are happening. An NIH-backed trial is testing phage therapy for Pseudomonas lung infections in people with cystic fibrosis, CF. That's a really tough infection to treat, so it's a big deal.
Can phages help antibiotics work better, not just replace them?
Yes, another exciting angle. Sometimes when bacteria evolve resistance to a phage, the mutation they make inadvertently makes them sensitive to an antibiotic again.
Right.
Like mutating an efflux pump makes it bad at pumping out drugs. Also, some phages produce enzymes, like polysaccharide depolymerase, that break down the biofilm matrix, allowing antibiotics to get in and work better. It's teamwork.
Wow, making old antibiotics useful again. That's huge. Are there other ways you can use phages or their parts?
Definitely. We can use phage enzymes directly as antimicrobials. Lysins, especially endolysins, are enzymes phages use to burst bacteria open. We can purify these enzymes and use them as drugs. They can shred bacterial cell walls very effectively, especially in gram-positive bacteria. And we can even use phages as delivery vehicles for CRISPR gene editing tools. We can load phages with CRISPR systems designed to target and cut out specific antibiotic resistance genes within bacteria. Deliver the CRISPR via the phage, snip out the resistance gene, and suddenly the bacteria is susceptible to antibiotics again. It's been shown to work against S. aureus and C. difficile in mice. Precision genetic warfare against resistance. Even beyond therapy, phages like crassphage are being used as markers to track human fecal contamination of water sources. So it really raises the question, facing this antibiotic resistance crisis, could phages, these natural bacterial predators, be a key part of the solution, offering sustainable, precise, and adaptable ways to fight infections? The potential seems enormous.
So wrapping this incredible journey up, what does this all mean for you listening? We started with the basics. What is a virus? Not quite alive, more like genetic code in a box, hijacking cells. We explored their ancient, maybe even primordial origins, challenging our ideas about early life. We saw their amazing diversity classified by structure and by their fundamental operating systems using the Baltimore scheme. We learned how they spread, subtly or obviously, and how that shapes their evolution. And then we completely flipped the script. We discovered viruses as beneficial partners, embedded in our own DNA, helping regulate our genes, helping microbes survive. And finally, we saw how we're harnessing them as powerful tools in gene therapy, as cancer killers, as eco-friendly pesticides, and as a potential answer to antibiotic resistance with phage therapy.
Exactly. To recap, this deep dive shows viruses are just so much more than pathogens. They're ancient evolutionary players, symbiotic partners, integral parts of our biology. And now, they're becoming sophisticated biological tools we're learning to use for incredible benefits. Our view is shifting from simple fear to complex understanding and application.
So maybe the next time you hear the word virus, consider this complexity. Consider that what we don't know about them, their hidden roles, their untapped potential, might be even more significant than what we do. They're not just disease agents, they're architects of life and potentially powerful allies.
Yeah, absolutely. Our evolving understanding just keeps reminding us that in nature, the lines between friend and foe, parasite and partner, are often way more blurred, more dynamic, and more interconnected than we ever thought possible.